Thank you very much for your introduction, and Rod, thank you very much for inviting me. It's a humbling experience to be here after some of these rock star speakers of the afternoon sessions. I'm not quite sure what I should do to grab your attention. So what I'll do, I'll talk about something completely different. I'm going to talk about something above the neck, about our brain, and about very severe mental illnesses, including schizophrenia, bipolar disorders, and major depression, and how nutritional, dietary, metabolic interventions actually start making a major headway in that area. So, but let's talk about the basics first. So what makes us to be able to think, feel emotions, move, to go out, grab a coffee and drink it? It's energy. It's energy in the form of adenosine triphosphate, or ATP. That's the energy-carrying molecule in every single cell of our body. If we do not have sufficient ATP production, we die. In fact, cyanide, the well-known poison, is an inhibitor of ATP production. After taking cyanide orally, the person probably have a minute and a half, and he will die. So ATP, without doubt, it's one of, if not the most important component of our life. Let's just look at the number. A single neuron in the human brain uses about 4.7 billion ATP molecules every second. And the reason I provided the map, because that roughly equals the whole population of the continent of Asia. So we are talking about one neuron every second. So this is something that has to be maintained every single second, possibly since conception until we die. So what does the brain use this tremendous amount of energy for? So if you think about the size of the brain makes up about 2% of the body mass, but it uses a disproportionately high amount of the body's total energy budget. It's up to 20%. And that is used for absolutely fundamental neurophysiological processes, such as maintaining the ability of neurons to get excited and then quiet down and communicate with one another. And beyond that, of course, ATP, and therefore energy, is needed to processes to synthesize molecules and to move molecules across the long processes of neurons. So in the human brain, a single neuron may have long processes called axons that are longer than 10 centimeters. That's a hell of a long way to go for a tiny little molecule, and that movement depends on adequate ATP provision. So now I think we get the picture. If something is not going quite right in the process that results in the adequate provision of the ATP molecules at the right place, at the right time, there are very dramatic functional consequences of that. This is the good old biochemistry textbook sort of stuff we all hated. I certainly did at medical school, and I never dreamed of actually working on it. So glucose gets into the cell with the help of insulin, as we all know in this crowd, and then very quickly phosphorylated by the enzyme hexokinase, just in order to get it stuck inside the cell so it can't get out because it becomes too polar. And then a cascade of enzymes break glucose down to pyruvate, and it produces 2 ATP molecules. It's a little bit of ATP, but it's very quick. It's actually a very quick production of ATP. And then it gets into the mitochondria, pyruvate, and gets into something that is called the TCA, or the tricarboxylic acid cycle, to produce reduced equivalent for a critical process in the mitochondria called oxidative phosphorylation. And that's the main site of action when most of the ATP molecules are being produced. So that produces a lot of ATP, about 32 ATP, from a single glucose molecule. So that's what maintains the generation of that dramatically huge amount of ATP. And on the side, glucose metabolism produces some other important things in neurons, including important neurotransmitters for brain cells to communicate with one another. And it also helps to generate DNA and RNA through a side process called the pentose phosphate cycle. So let's focus on this bit here, the ATP production, and think about the link between metabolism in the body. That's the sort of thing we have been discussing at this meeting so far, and then what's happening in the brain. And it's always really interesting to look at the historical context of it, because, we think we just came up with a great idea, but as it turns out, it's been discovered for a long time, and it's been known for a long time. So Thomas Willis, the great physician and anatomist, back in the 17th century in Oxford, he described the basic circulation of the brain, and the circus of Willis is named after him. So he was the first one, actually the first one we know about, who realized that diabetes often appeared among people who had experienced stressful life events, sadness, or long sorrow. So that was the first link between something that's going on in the brain and something that otherwise seems to be in the domain of the rest of the body. But that later on in the 19th century, the famous British psychiatrist Henry Maudsley made some really interesting observations. He observed that diabetes seems to be happening in a much higher frequency in people, in the families of people with insanity, which roughly translates to our diagnosis of schizophrenia today. So that was a very pertinent observation, which then was supported by some early experimental data. So this is sort of mid-20th century glucose tolerance test we all know about. So that's the normal one. Glucose comes in, goes up, and then if insulin production and insulin response is adequate, then it should go down. People with diabetes, back in those days, not much drug therapy against it. They had this profile, so glucose did not return to the baseline because of that developing insulin tolerance, of course. That's not the surprising bit. The surprising bit is here. So people with schizophrenia showed a glucose tolerance profile that was indistinguishable from the ones in people with diabetes. At the time, insulin shock was actually used in the treatment of a number of psychiatric disorders, not because they had any sort of mechanistic insight why it should work. It was purely by experience. People after coming out of the hypoglycemic coma, they felt better. Whatever that first. But that's not the important. The important point here is that they discovered that people with schizophrenia actually required a lot more insulin to push them into a hypoglycemic state, which again strongly suggested that there might be an insulin-resistant state at the background. So this is a paper we published some years ago, actually showing exactly that. An increased insulin resistance in people with schizophrenia, not in people with major depression. And of course, Jessica alluded to the link between stress and elevated glucose. So you can make the argument that, yeah, these people were just freshly hospitalized, they are acutely psychotic, therefore they are highly stressed, and that drives up their glucose and impairs their insulin resistance. So we actually try to control for that, and and it didn't seem to be the case. So none of the stress biomarkers correlated with their glucose homeostasis at all. So it seems to be an inherent phenomenon related to schizophrenia. These are first episode non-medicated patients, and that's important to emphasize. A relatively recent systematic review and meta-analysis put all of these together into beautiful, beautiful analysis showing consistently that people with schizophrenia, never medicated, first episode, they have elevated circulating blood glucose and they have insulin resistance. So let's look at what's happening in the brain. What you see there is images emphasizing decreased frontal cortical glucose metabolism. Again, in never medicated schizophrenia patients. And I'm using the never medicated all over again because we know that antipsychotic medication wreaks havoc on glucose metabolism. It increases blood glucose. Patients develop insulin resistance and they gain tremendous weight very quickly. So that's why I'm emphasizing that these are non-medicated individuals. And this is one of the most consistently replicable findings in biological psychiatry: hypofrontality, decreased metabolic activity of the frontal cortex in schizophrenia. A more recent data from Harvard showing abnormal storage and use of brain energy, and it seemed as if ATP production from that mitochondrial ATP production switches to glycolytic ATP production, as if mitochondria would not be able to work properly, and the cells are desperately trying to compensate for that. At the same time, post-mortem studies show decreased amount of glycolytic enzyme proteins, decreased expression of genes encoding for glycolytic enzymes. Again, these are from isolated neurons. These are not pieces of brains. These are actual isolated, characterized neurons from the brain of people who died while having schizophrenia. So now we moved on to that tiny little organelle in our cell called mitochondria. So mitochondria are quite unique. They used to be bacteria back in the very, very old days, about 1.5 billion ago, and they were then fused by other prokaryotic cells, and they carried the great advantage of being able to use use oxygen to generate ATP. And that was the time at the history of the Earth when the oxygen concentration started increase in the atmosphere, actually very significantly. And we all heard about oxidative damage. So the tiny little unicellular organisms that did not have the mechanism to deal with the elevated oxygen concentration died out. The ones who had this tiny predecessor of the mitochondria in them, they were able to survive and not only use the excess oxygen, but to generate something terribly helpful, and that was ATP. So if we look at that metabolic process in the mitochondria, oxidative phosphorylation, you see that a number of different elements of this process seem to be abnormal, not functioning, differentially expressed, impaired in schizophrenia and bipolar disorder. This is a very recent study, just published earlier this year, late last year, specifically identifying a particular type of neurons, nerve cells, called GABAergic inhibitory interneurons. So those are the ones that tell all of the excitatory cells when to shut up. So they are really, really very important. And in this study, they identified that the most significant differentially expressed genes in individuals with schizophrenia were the genes that encoded for oxidative phosphorylation and other energy producing processes in the mitochondria. So we try to make sense of that in a recent paper in Molecular Psychiatry and identifies schizophrenia as a disease of broken brain bioenergetics. So besides the fact that it alliterates and sounds good, it seems to be something that is really going on. And one way of proving that is what happens if we try to normalize that broken brain bioenergetics. And that gave rise to the idea of using an alternative source of energy that does not at all rely on glucose provision. Therefore, it does not rely on glycolysis, which seems to be impaired in the brain of individuals with schizophrenia. And beside that, it carries a number of other potential advantages, such as restoring oxidative capacity of the cells, replenishing the missing acetyl coenzyme A to feed the TCA cycle and all that. So that initiated some preclinical work in my own lab about 8 years ago, and we published a number of papers, all in mice, so these are not human papers, in which we created different animal models of schizophrenia and used ketogenic diet to see whether they can be normalized. And across the different models, we have unequivocally positive results, showing that being on ketogenic diet prevents these animals to develop schizophrenia-like behaviors. And all of these are published, so I don't want to bore you with the details. I just want to show you one interesting set of data which we have generated and we haven't yet published. And it's based on one of the most consistent sort of etiological factors of schizophrenia, which is called the maternal immune activation. There's a lot of data from many different countries, a lot of epidemiological data linking viral or even bacterial infection during second trimester of pregnancy with the later emergence of schizophrenia. Which doesn't mean that if someone has a flu midway through the pregnancy, the child will have schizophrenia, but it increases the likelihood about twofold. So it's a significant increase. So we modeled that in mice, did exactly that. The details are not important. What's important is that we can generate a behavioral feature of these animals that are not identical, of course, but has parallels to what people with schizophrenia have: increased repetitive behavior, decreased sociability, an impairment of a very basic sensory motor gating process, and impairments in working memory. With ketogenic diet, we can normalize all of that. And we have done a lot more studies in that line, but let's just move into what you are probably more interested in, what's happening in humans. So starting of 2017, there have been a number of case studies published showing great effect of ketogenic diet in people with schizophrenia and schizoaffective disorders. Case studies are great. They show you indication that something might be happening, but it's a one-on-one relationship between that particular patient responds particularly well. So how much can you generalize from that? It's always limited. So we need a little bit more proper studies with adequate number of individuals included. So this is an example to some of those studies. These are 31 individuals with severe and treatment-resistant psychiatric disorders, including bipolar disorders, major depressive disorder, and schizoaffective disorder. They were put on ketogenic diet for a different lengths of time, and they showed tremendous improvement. These are chronic patients, treatment-resistant, have been ill for a long time, have been on medications of different kind, a whole dozen of medications. And you can see they did not only improve in their clinical general impression, that's their general well-being, but they were able to cut back on their medication, which is really important because those medications are unfortunately metabolically not very helpful. This study just came out actually literally early this week by a group from the University of Edinburgh who just recently completed this study using ketogenic diet in individuals with bipolar disorder. This is not a randomized controlled clinical trial, by the way. It was just single arm, and they knew that they were on ketogenic diet in the hope of getting better. Having said that, first of all, they were able to comply with the diet. 26 commenced and 20 completed. And we are talking about mentally ill individuals who have serious difficulties in many aspects in their lives. 95% of them were able to adhere to the diet. They were able to keep up a decent blood ketone level throughout the study, and normal glucose readings. So all those hyperglycemias seem to be disappearing. Very importantly, from the psychiatric point of view, there was a very good correlation between their blood ketone levels and their symptomatic improvement. So they did not only improve, but that improvement seemed to be related, whatever correlation actually means, but it seems that increasing blood ketone was related to their symptomatic improvement. One patient in the cohort especially had high increases in her blood ketone, and she happened to show the strongest improvement. They were also interested in looking into what's going on in the brain of these individuals, so they did magnetic resonance spectroscopy analysis of different neurochemicals in the brain. And what they picked up was the main excitatory neurotransmitter in the brain, glutamate. So 80% of all of our neurons in the brain are glutamatergic neurons, and glutamate seems to play a role in a number of psychiatric disorders. As a result of being on ketogenic diet, these individuals had a drop in their brain glutamate level. Whether that's related to their symptomatic improvement or not, we don't know, but this is the very first piece of data anyone has ever seen in relation to ketogenic diet in psychiatric disorders and something going on in the brain. Another recently completed study done by Shibani Sethi at Stanford University with people who had schizophrenia and bipolar disorders and also had metabolic abnormalities associated with that. The data I'm showing to you are unpublished data shared by Shibani for the purpose of me able to show that to people in the conference. A lot of data, and I'm just going to highlight some important aspect. If we look at the psychiatric changes, these people improved on ketogenic diet. Many of these patients in this particular trial were not actually located in Palo Alto, California. So they did not attend physically a clinic. So it was mostly through remote correspondence and trying to make sure that the diet is being properly followed. Shibani worked with the patient's local doctors. And even under these quite challenging circumstances, these individuals showed great improvement in their symptoms. But not only in their symptoms, but also some really appropriate biomarkers, such as the inflammatory marker, high sensitivity, C-reactive protein, decreased, blood lipids. So this is not the crowd I need to convince, but I think outside of that room, I would have a very hard time to convince the medical establishment that if someone is on a ketogenic diet, we are not necessarily killing that person by stuffing up their arteries with fat. And interestingly, again, these are individuals who started out with a very, very bad metabolic background. They ended up having decreased triglycerides, increased HDL cholesterol, and you will see they all lost weight. Their BMI became much better. In the next slide. One other aspect I would like to emphasize. So some of the participants of the trial were able to follow ketogenic diet quite religiously, having been in ketosis throughout the whole trial. Those are the adherent ones. Some, on the other hand, went on and off from ketosis. So they were the semi-adherent. And interestingly, we are seeing something almost like a dose-response effect. So you get a bit of an improvement with partially adhering to the ketogenic diet, and you see a much bigger improvement in individuals who properly maintained ketosis over time. So this is really exciting. So if any one of you ever dealt with people suffering from eating disorders, anorexia nervosa in particular, you know that they are very obsessed with their food, and I would have been the last to imagine that this is an area where a very strict, highly prescribed dietary therapeutic approach would have effect. And in fact, just very recently, a paper was published in 5 individuals, adults, who actually recovered from, in terms of their BMI, recovered from eating disorders, but were still very much obsessed with food. They were put on ketogenic diet for 4 weeks, and then they received 4 episodes of ketamine injections. So it was a combined treatment. In order to get rid of the potential effects of ketamine, you just focus on that line going through the figures after the first data point. So that was the end of the ketogenic diet period before they got their ketamine. And even that 4 weeks on ketogenic diet resulted improvements in their attitude towards food, in their body image. So this was very surprising and really exciting set of data. So that brings us to our own study. So we are just about to start the world's first randomized controlled clinical trial in which we compare the efficacy of ketogenic therapy with another dietary intervention in a sense. That's the standard healthy Australian diet, which is likely to be much superior to the diet our patients would be otherwise eating. So we are just about to start after a long journey through human ethics and administration. We just last week got the site-specific approval from Townsville Hospital and the university, pretty much ready to start. We are going to recruit 50 bipolar and 50 people with schizophrenia/schizoaffective disorders. There will be 2 arms, the ketogenic diet and the Australian dietary guideline Eat for Health for 2 weeks. And we are interested in efficacy of the dietary intervention. And we are going to measure that by using a number of psychiatric rating scales, as well as highly validated and quite extensively used, especially research purposes, cognitive testing battery, because cognitive impairments, especially influencing the prefrontal cortical functions, are quite prevalent in both schizophrenia and bipolar disorder. And then, of course, we are going to look into a number of other measures, which we will integrate through using artificial intelligence and machine learning, and that will include the use of wearable devices to continuously monitor heart rate variability, skin temperature, skin conductance, and activity over a period of a week prior to and at the end of the dietary trial. And the big challenge we are having, besides recruiting and running the trial, is actually integrating the data from so many different domains. And that's why we are collaborating with a group of engineers at my university who are experts in machine learning and artificial intelligence. We are lucky to have an extremely strong team. I'm working with a senior psychiatrist in the hospital, Carlo Longitano, in setting up and running the trial. And we have a team of colleagues, including endocrinologists, very importantly, a senior dietitian who has long experience in using ketogenic diet in many different medical settings, epilepsy, gastrointestinal conditions, and all that. So putting all of that together, at the moment it seems, on the basis of the available evidence, none of those are randomized controlled clinical trials yet. I have to emphasize it. But ketogenic therapy seems to show great promise in a wide array of severe mental disorders. Thank you very much.